All right, sounds good. Let's get started so we don't waste time. Welcome to day 2 of the library for NGS protocol. I'm happy to see that the majority of you guys are back. Hopefully you did a good job yesterday. If you have questions from yesterday, you know, you have time to ask them today as well, and also tomorrow. If you have specialized project you want to talk about, just shoot me an email, and then we can discuss. Okay. Housekeeping notes, same as yesterday. So no questions in the chat. Just use the chat either to answer my questions. So again, the polls function unfortunately is not available this time. But feel free to answer the questions that I'm asking you on the chat. And also, if there are any problems with the audio video. First question, can you hear me? Can you see the screen just fine? Okay. And let me share the Google Sheet link again. So it's the same as yesterday. Okay. We're gonna try to do the same as yesterday. So if you can just quickly check if you can access the Google Sheet and try to type something, maybe in the questions from yesterday, you know, just a letter just to see if you can write something, and just let me know. So we're going to proceed in the same way. The class is being recorded already, and you know, just make sure your mic is muted. I'm not sure actually if you can. Okay, awesome, you can edit. Great. Perfect. So let's get started. Again, today we're going to talk about some specialty DNA library preparation. So it's basically just variations on the theme of the library preparation that we did yesterday. And we're going to focus today on the RNA library preparation. And at the end, we're going to talk about some best practices for NGS, which I think are really important, but not many people are using them actually. So this is basically the layout of today. So we're gonna do like a small summary from yesterday. Then we're gonna switch gear to specialty DNA sequencing libraries, and then we're gonna talk in details about the RNA library preparation, stranded and unstranded type of library preparation. Also, the point 3 is, I think it's very important, especially for RNA, how to do the RNA QC and what is the RNA integrity. And we're going to briefly touch on the low input single cell RNA-seq methods. Couple of slides, maybe even one, on the small RNA-seq approach, and then again, best practices in NGS. As you can see, we're trying to do Kahoot again. I hope you enjoyed it yesterday. If not, I'm sorry for you. And then we're going to do 2 mini quiz and attendance. So the quizzes, again, they're not for grading. However, they're really important. So I'm taking attendance through the mini quiz. So if you don't fill in the mini quiz, you will not get credit, okay? And I'm using this mini quiz if you have problems in the final quiz of tomorrow. So remember, tomorrow we're going to have a mini quiz. Sorry, a final test. My bad. So if your score is quite low in the final test, I will come back to the mini quiz and see if you did well or if you didn't do very well. And then I'll try to see if I can increase your grade a little bit just by using the mini quizzes, okay? All right, so if you remember yesterday, we basically discussed about these library preparation, and what we've done yesterday is how to measure the DNA, okay, And as you remember from yesterday, we used two methods to measure the DNA. One, which is the nanodrop, which is a spectrophotometric method, is giving us the presence or absence of contaminants. Okay, so the ratios 260 and 280 for the presence of protein or also the RNA contaminants, or 260, 230 for presence of organic, such as phenol, such as carbohydrates, and so on and so forth. So once we've done the concentration with the qubit again, so the nanodrop only for measuring the quality, the concentration we usually perform with the qubit. Then we go ahead and do fragmentation first, because our fragments in the final library needs to be short. And then we perform end repair and A tailing, which basically modifies our fragmented DNA in a way that it's compatible with the adapters that we're using. And so after end repair and A tailing, we can perform adapter ligation. And now we have satisfied all the criteria for sequencing. And then we have our final library, okay? I just want to point out, I don't know if you noticed this little difference, but actually after we perform adapter ligation, that's a final library, okay? So the PCR is actually not needed for like 100% of the times, okay? So that's something that we can do afterwards, okay? We only need the PCR if we want to increase the number, the amount of our final library, or if we have to add the P5 and P7 and the barcodes that were not present in special adapters, okay? But usually the adapters that these preps are using are full length, okay? So full length means that they have all the information that is needed for sequencing, okay? After we finish our preparation with or without PCR, we measure the concentration, and then we do the QC. So if you remember the concentration, we usually measure with the qubit, but you can also measure the concentration through a qPCR method, okay? So the qPCR method is basically taking advantage of a standard curve that you create with some standards that the kit that you're using is usually giving you. And then you perform dilutions of your final library, and then you do a qPCR. And then you compare your CT values against the standard curve that was created using the standards. And so just by doing this comparison, you can basically understand what is the concentration of your final library. Also very important, the QC, the final QC. Again, you can use agarose gel if you don't have a TapeStation or bioanalyzer. But again, I hope I made a good point why you want to use the TapeStation or bioanalyzer, because we want to see the presence of a very minute amount of contaminants, which will make our life very difficult if we use just the agarose gel. Okay. All right. I hope this is clear. And that's exactly what I was talking about. So library A, B, or C. You see in A, we have PCR primer dimers. In B, we have adapter dimers. And in C, we have a clean library, no contaminants in the regions that we expect them, which are basically between 50 base pairs to 150 base pairs. And the reason why we want to look them in detail is because the amount of these primer dimers and adapter dimers, it's better if it's below 1%, okay? All right. Again, this is very important, how the sequencing work. This is something that usually the sequencing core is asking you, okay? So what do you want me to do with your libraries? So you have to be able to tell them, I want to sequence my libraries in a certain way. And then you have two modes, okay? You have the insert mode, okay? So this is one. We can just call it A. Sorry for my handwriting. And then also you have the indices mode, which is a second mode of sequencing, okay? we can call it B. Okay, so for A, okay, so for the sequencing of your DNA insert, you can do single end, and in this case, it will only be the pink primer that it's described as read one, or paired end, which is both read one and read two in green. Okay, paired end, one more time, means that your DNA insert is sequenced from both ends. Okay, so from one end and from the other end. Okay, so this is a sequence modality A, and then you have to tell the core, okay, so you sequence either single end or paired end. Then what about your indices or barcodes? Okay, you can have a single index. Okay, so single index will sequence the index 1 or I7 that is situated on the P7 adapter, and a dual index will sequence both index 1 and index 2, which is also called I5, which is in dark red here in the picture. Okay, I also have some examples here. What you usually sequence with a single end 50, RNA-seq by sulfate-seq for single end 100, paired end Ataxic high-seq, and sometimes ChIP-seq. I have to make this modification. So also, you can sequence paired end 100 or paired end 150, and this is for RNA-seq if you're doing specific application, or usually when you sequence DNA. And the reason why we use these long reads is because the cost per base, is cheaper. Okay, so that's why we are using longer reads for these applications. We just basically they just require a lot of sequencing depth. Okay. What else on this slide? Oh yeah. So if you notice the application that are mentioned here, single end 50, single end 100, paired end 50, and paired end 100. They're only mentioning you know, the insert mode, because the index mode or the barcode mode, it's completely independent on the application. Okay, that depends on the adapters that you have. Okay, so just want to make sure you understand the difference between the two sequencing modes that you can request. Okay, and today we're focusing on the RNA. But again, before we are doing some specialized library DNA library preparation, and so we are talking about ChIP-seq, we're talking about bisulfate-seq, we're talking about Ataxic, and that's it. Okay, so these two guys, we did it yesterday. Okay, so we're not doing this anymore, and we're not treating Hi-C. It's a very complicated protocol. I don't want to. It's not very common, but you know, if you want to know more about it, I'm happy to discuss that in private with you. Okay. All right. So let me check quickly if you have questions from yesterday. And let's wait a couple of minutes. Doesn't seem like it, but let's see. Let's wait a couple of minutes. Doesn't look like anybody has anything to ask. Let's go ahead. Okay, so today we are talking about specialty library preparation. Okay, and these are variations on the DNA library preparation. So the first one that we're talking about, it's actually my specialty. So I'm specialized in DNA methylation library preparations. But I've done many others. It's just like DNA methylation was my first love, and so DNA methylation, just a biological function behind it. it's a covalent mark in cytosines. In mammalian cells are usually CpG sites that are methylated. Um, uh, and just asking, answering from Mohammed, yes, I'm recording today's class. I'll give you the link once it's ready. Um, going back to the methylation, it's a covalent modification on CpG sites in mammalian cells, and it's it's correlated to gene expression if it's in the promoters. It's also correlated with gene expression if it's in the gene body, okay? It's just like usually when the promoter is methylated, it's anti-correlated, okay? So the gene is usually not expressed. When it's in the gene body, usually the gene means it is expressed or it has been expressed in the past. It's a really cool modification because it does not modify the sequence of the DNA, but it's different between cells. So it's we actually use DNA methylation to distinguish cells from a population of tissue. That's why we study DNA methylation. Yes, the video of yesterday was recorded, and it will be available, sometime soon. Okay, so it will be uploaded in the collaboratory website. So how do we prepare a library for DNA methylation? So we actually perform exactly the same steps as before. So we have double-stranded DNA. We do fragmentation and repair phosphorylation, A-tailing, and then we add the adapters, okay? And so this is just a regular library preparation. However, the adapters that we're actually adding right here are slightly different to the adapters that we saw yesterday. Not in the sequence itself. The sequence is exactly the same. They still have to go on an Illumina machine. However, the adapters are pre-methylated, okay? So that means that basically all the cytosines that are found in these adapters they carry the cytosine methylation modification, okay? And the reason we do that, is because we are using this chemical. It's called bisulfate, okay? It's a chemical that is basically modifying all the cytosines into uracil unless they are methylated, okay? So if you look on the box, or the dotted box, blue box on the right, you see an example, okay? So this is a DNA fragment, okay? So in this DNA fragment, you have a thymine, you have a cytosine that it's not methylated, you have a thymine that, and then you have a methylated cytosine, and then a guanine, okay? So I'm looking at this filament right here. After the bisulfate treatment, the cytosines will be converted into uracil, okay? And the methylated cytosine will stay methylated cytosine, okay? After this treatment, we have to perform PCR, okay? And in this step, uracil is read, by the polymerase as if it were a thymine, okay? So after PCR, it gets transformed into thymine, okay? So you have thymine, 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 and then the methylated cytosines will stay cytosine after PCR amplification, okay? So this is just the bisulfate conversion. So it will convert all the cytosines unless they are methylated, okay? So that's how it works. And we perform the bisulfate conversion after the adapter ligation, okay? So if you imagine that the sequence on the adapters has to be the same from the adapters that we used yesterday, if we have cytosines in the adapters and they're not methylated, the bisulfate conversion will convert cytosines into uracil and then into thymine after PCR, which means that the sequence of the adapters will vary, okay? So the only way to maintain the same sequence of the adapters after bisulfate conversion is to have pre-methylated adapters, okay? And so that's why we are using these special adapters in this case. The sequence is the same, it's just like they're modified with methylation, okay? Another note, after bisulfite conversion, the final product, it's a single-stranded DNA and it contains uracil, as I was mentioning before. And not all of the DNA polymerases they can read uracil. Sometimes the polymerase, and we'll see this in this stranded RNA-seq approach actually, some polymerases are not able to read the uracil, they stall on the uracil, so they're not able to amplify the fragment, okay? So in this case, we're using either Taq polymerase, which is able to read the uracil, or engineered polymerases that are, that have been modified to recognize the uracil and be able to amplify a uracil, okay? Again, this is a very basic DNA methylation protocol. There are many variations of the protocol. So if you're interested, if you have a project with DNA methylation, please talk to me. I can point you out in the correct direction, what type of approach to use. All right. Let's go ahead. Okay. So who knows what chip sequencing is? Who wants to type in the chat what is the purpose of chip sequencing type of approach? Let's see if we got some answers. Perfect. So I got an answer from Jonathan. Identify DNA bound to specific proteins. That's correct. So basically, the ChIP-seq stands for chromatin immunoprecipitation coupled to sequencing. Okay. So what you usually have is, you're very interested in a protein that in this case binds to the DNA, but you want to know where in the genome this protein binds. So for example, a transcription factor, that you want to know where this transcription factor is binding, for example, to regulate, genes, maybe activating those genes or, repressing those genes. And so this is a way of basically understanding where your protein binds on the gene in the genome. Okay, so how the protocol works, it's very simple in terms of like a theoretical explanation. It's actually very difficult to perform. You might have a lot of problems with ChIP-seq type of level preparations. The reason is because you are not starting from naked DNA, okay? You're starting from chromatin preparation, okay? So you basically extract the chromatin from the cells, or what you can do is you treat basically the cells with a cross-linking agent. Most of the times, it's formaldehyde, or you can have other cross-linking agents. They have longer arms, but basically the proteins and the DNA that are close to each other, they're basically covalently cross-linked. Okay, so they're covalently attached to each other. What you do next is you basically fragment your sample. So you fragment your chromatin either by using sonication or by using endonucleases, such DNase 1 or MNase. There are many specific endonucleases. And after these digestion or fragmentation, you can call it fragmentation if you want, you incubate your fragmented chromatin with antibodies that are recognizing your protein of interest, okay? So for example, up top here, you can see this guy was interested in histone modifications, okay? So you can recognize that. While in this case, this group was interested in a protein complex. So there are basically antibodies against this protein complex. And so once you do this immunoprecipitation, you basically wash out all the DNA that does not have our protein or histone modification of interest. and then you basically isolate the DNA that was originally bound to your cross-linked chromatin, okay? What you have to do between the after immunoprecipitation to get pure DNA, you basically have to decross-link, okay? So you basically have to remove these covalent binding that you created during the formaldehyde treatment, and then you have naked DNA that you can add the adapters to, okay? So if we do a parallel with the regular library preparation, you remember the library preparation starts with the fragmentation and repair phosphorylation, A-tailing, adapter ligation. However, in this case, we already performed the fragmentation, okay? So our fragments are already short to begin with. Wow, where is my mouse bone? Okay. After you do DNA purification after the IP, you don't have to fragment anymore. You can just go ahead and perform and repair phosphorylation, A-tailing, and adapter ligation and PCR, okay? Usually in this case, PCR it's needed just because you usually don't have too much material after the immunoprecipitation, okay? We are talking about like few nanograms. I mean, it really depends on the protein that you are pulling down. For some proteins, for example, for histones, unmodified histones or a very common histone modification, you might have tons of DNA after immunoprecipitation and purification. For some other modifications, such as transcription factors, for example, you might not have a lot of DNA. Sometimes you're not even able to quantify the immunoprecipitated DNA, and not even with the qubit high sensitivity. Okay, so what I usually do when I perform a ChIP-seq experiment, I try to quantify, but even if I don't see anything, so even if the qubit is telling me, okay, your sample is too low in concentration, you might have something, you might not have anything. What I usually do is I go ahead and prepare the library, and just by doing amplification with the PCR at the end, I usually increase the amount to a lot of nanograms, and so at that point, I have a lot of material which I can test. So what you usually do before sequencing your chip sequencing experiment libraries, you usually perform a qPCR on some loci that from the literature, or from other groups that are bound by your protein. So you design qPCR primers, and you basically test the enrichment score. So you basically do the qPCR on the IP DNA and on the input. But again, you should normalize the amount of DNA before performing the qPCR. Otherwise, you cannot compare the two qPCR results. And you can do that on your final library, okay? So if you're not able to quantify your DNA after immunoprecipitation, you can prepare the library, and then you can perform the qPCR on your final library. Usually, from my experience, the enrichment decreases 2-3-fold, okay? So for example, if I have an enrichment of, let's say, 10 here, actually in the original IP, I usually have around like 50-100, okay, of enrichment. But if you don't have too much material, that's the only way to just try the qPCR before sequencing, okay? A very important part of the chip sequencing experiment is actually the sonication and fragmentation and the antibodies, okay? So these are the two steps or two reagents that are really, really important, okay? So the sonication, it's really important because you want to fragment your DNA into a specific size. I've told you, usually the fragments that are sequenced by the Illumina are like between 200 and 800 base pairs. So you want to have your fragments in that range. However, if you sonicate too much, you will get the fragments in that range, but you can also destroy the epitopes of the protein that you want to pull down, okay? Remember, you're sonicating, you're doing fragmentation on the chromatin, okay? So it's both DNA and protein at the same time. And that is before immunoprecipitation, okay? So before adding your antibody. That's why many groups are using endonucleases for fragmentation. They're much gentler on the chromatin than regular sonication, okay? The other key point is the use of the antibody, okay? So if you don't have an antibody that recognizes your protein, you are welcome to develop your own. It will take you like a long time, months. However, if there are like antibodies that exist for your protein, just make sure they are ChIP sequencing grade, okay? Not all of the antibodies are ChIP sequencing grade. So just make sure that your antibody is of a good grade for ChIP sequencing. Otherwise, your results will be like super crappy, and basically you're not going to be able to interpret them, okay? Another application that I want to talk about, it's called Ataxic. So it's assay for transposes accessible chromatin. So what this is doing, it's basically, again, we are starting from chromatin, okay? So in the past two applications, ChIP-seq and Ataxic, we're not starting from purified DNA. So if you're, for example, extracting these material and you're measuring at the nanodrop, you expect to have a lot of protein contamination, okay? That's normal. But basically what happens is you add these T5 transposon to the chromatin, and this transposon is actually cutting the DNA and adding the adapters only when the DNA is not bound by a protein, okay? So if DNA is bound by a protein, like for example in nucleosomes or in regions where you have a huge occupancy of transcription factors, in that region, the transposon is not able to cut the DNA, okay? If you remember yesterday, actually, I talked about an application where we use exactly the same transposon, okay? That was the Nextera type of library preparation, okay? The so-called tagmentation, okay? And in that case, though, we were using this enzyme on naked DNA, while here we are using this enzyme on the chromatin, okay? The information that the addition of this enzyme on the chromatin is giving us is telling us, okay, so is this region open or closed, okay? So is this accessible to transcription factors or not? Usually the open regions in the genome are considered to be active in sense of transcription and regulation, while the closed regions are considered to be inactive or repressed, okay? So if you look at the plot at the bottom, okay, so that's a genome browser view. You see that basically when you have an Ataxic peak here, it means that this region is open. So it's accessible to this transposon, okay? And if it's open, most likely the gene that it's next to it, or the gene that it's, the gene that is next to it, it's usually transcribed, okay? And as you can see, so if you basically go down Ataxic, you see a peak here. You see also here some peaks that are basically H3K4 trimethylation, H3K27 acetylation, and H3K4 monomethylation. And these are all marks of active chromatin, okay? So it makes sense with the fact that we find a peak of that is accessible to this transposon, okay? So how does a Ataxic library look like? It's very different from our regular libraries. We'll see in the TapeStation, a view of the TapeStation, but just for now, basically disregard these purple and green peak, which are actually green and purple here. I'll explain that to you tomorrow. But basically, you don't see like a smear that we are used to see in our final libraries. You see actually a ladder, okay? And the ladder is actually is reflecting the chromatin that we are capturing, okay? So the black one, the black region here, it's called subnucleosomal DNA, okay? So this is the size of the fragments that are smaller than a nucleosome, and they're usually just stretches of open chromatin. Okay, so if you look on the left, the black one is here. You actually see that the black part is bigger than the other ones, just because it's completely open. So the enzyme can just cut anywhere, and that's why you don't have a big chunk of DNA. It's just like very fragmented. So it can be here, 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 or here. Okay, it can be anywhere. So that's why you have very short fragments. Okay. Then if you go on the red one, this is a mononucleosome. Okay, and the size, it might not make sense for you right now, but if you look at the size, it's like around like 300. Okay, so question: how big is the DNA wrapped around nucleosomes, more or less? You can just type it in the chat. You know, just a ballpark idea. It's like 100, 1,000, 500, 150. Okay, so it's yeah, it's close to it. It's usually like 160, 170. Okay, and the sum of the adapters, the length of the adapters is actually 130, 140. Okay, if you remember, the adapter diameter was exactly this size. Okay, so if you sum up 130, 140 to 160, 170 of the mononucleosome, you end up with 300, which is exactly the size of the mononucleosomal final library that we see here. Okay, so it's mononucleosome red, and then we have dinucleosome blue and trinucleosome green. You can see on the left you have one nucleosome, and then you have two nucleosomes here, and then you have three nucleosomes here. Okay, so this is the size of the library that we capture. Okay, again, ATAC is just you see basically an enrichment of reads over the regions that are basically open. So there's no protein bound to it. Okay, and depending on how big is the region and how open is the region, you can see like a broader peak or a tighter peak. Okay, and most of the times you will capture the nucleosome signal. Okay. All right, there are variations on this. I know that Ataxic is a very popular application. So the original paper was described quite a long time ago. There have been major modifications to actually minor modifications, but with the major improvement of the output to the technique. You can do single cell if you want, and also the new technique for Ataxic, it's called Omni-Attack, which is also good for frozen tissues, for example, and it will also reduce contaminant read that was coming from the mitochondrial DNA. Okay, so mitochondrial DNA with the original Ataxic type of library preparation was a big issue. Usually you have about 60, depending on the type of cell that you're performing your experiment, it can vary between 40 to 80% of the reads. And usually nobody cares about mitochondria. They're not supposed to be, they're not supposed to have chromatin anyway. So usually you don't assess the chromatin status of the mitochondria with the transposase. So usually it's just like wasted reads. However, with this modification, the Omni-Attack, you are able to get rid of this mitochondrial DNA contamination or reduce it greatly, up to 20% maximum. So you're not wasting too many reads. And again, I told you you can perform this experiment on frozen tissues as well. Okay. All right, and all right, let's take a look at the Google form first. Sorry, Google spreadsheet first, just to check if you guys have questions, and then we are gonna do a quick mini quiz. Okay. Okay. See, there might be a question. Okay. just want to remember everyone that, okay, how much mitochondrial DNA contamination if the sample is from tissue? Well, it really depends on the sample. I mean, I cannot tell you, it depends on the preparation of the sample. Also for Ataxic, I didn't mention it in my slides, but dead cells are a huge issue, okay? So usually they recommend to have a very limited amount, up to 5% of dead cells. If you have more, your signal will not be great. You basically see signal everywhere without seeing too many peaks, and that is a huge problem. Okay, so I have to add it to my slides. Both mitochondrial DNA contamination and dead cells are a huge issue. Okay, if you have a lot of dead cells, um, you have to try to get rid of those. I don't know, fact sorting, or if there are like some magnetic beads that can get rid of it. You can also try to perform DNase 1 digestion on intact cells. And so DNase 1 is basically a endonuclease that is degrading all the DNA. But if you perform that on, on the cells, it will only degrade the DNA that it's extracellular. Okay, so we'll get rid of the DNA from dead cells. Okay, but yeah, dead cells are, I would say, if you have a tissue, dead cells is probably a bigger problem than mitochondrial contamination. But again, if you're using this new protocol, the Omni Attack, the mitochondrial, they're basically using a different detergent during the lysis of the cells. And so this different detergent is not basically, it's getting rid of the mitochondrial DNA contamination. Okay, so I think if you have tissues, then you have to worry more about the dead cells. Okay, let's wait one more minute. Or if I don't see any questions, we can let me get the link to mini quiz 3, and so I can share it with you. And so I'll give you 5 minutes. So we reconvene at 2:25-ish. Okay, let's do, why don't we just do like a 12:30, so we can have a little break as well. Okay, so we come back at 2:30 with the mini quiz and a small break for hydration. All right. Okay, we're back at 2:30. Let's go back to the workshop. Can you hear me just fine? Please type it in the chat because I removed my. Okay, perfect. Okay, so now we are gonna start to talk about the RNA. Oh, by the way, I saw that a couple of you didn't answer the quiz. I just want to make sure you understand this is not only the quiz, but it's also attendance. So if you're not signed up with the quiz, I don't care if you got like 0 points, but if you're not signed up, you will not get credit for the attendance. Okay, just want to make sure of that. Okay, so let's go back to our DNA schematic. So now we are dealing with RNA. Okay, so let's pretend if I'm genomic DNA, my wife is RNA. Okay, question. So what is the the main difference. So of course we don't have gDNA. Okay, so how do we transform the RNA into DNA, which is one of the few features that the Illumina requires? What would you do? And you can answer in the chat. Okay, I don't see any reverse transcription. Perfect. Yes. Okay, awesome. Yes, so we're basically converting the RNA into cDNA using reverse transcriptase, and this step, it's called reverse transcription, of course. So that was a good answer. And so what we're going to do is we're going to perform reverse transcription, okay? However, we are not performing reverse transcription at the very beginning as the first step for one main reason. Because we have different types of RNAs in our cell, okay? And the question I want to ask you is, do you know what RNA is the most abundant in our cells? Yes, exactly, ribosomal RNA. Do you know more or less how much was the percentage of the ribosomal RNA that we have on average in our cells? Yeah, so it varies between 70 to 90%. And do you know what is the percentage of messenger RNA, which is usually the molecule that we want to assess the transcription levels? So messenger RNA, it varies again. It's usually about like 5%. Okay, so it's a very minor amount of the total RNA that we have in our cells. Okay, so what we usually do is we do a so here are the percentage. Okay, so ribosomal RNA, the one that we just talked about, it's about like I would say like a 70 to 90%. And then we have abundant non-coding RNAs, such as tRNAs, for example. They're like a 10-15%, so it's usually a lot. But these tRNAs are much shorter in size. Okay, so they're usually not incorporated into the library preparation. And then we have messenger RNA, which is, on average, you can think of it like as 5%. And then we have, other type of RNAs, such as long non-coding RNAs, SNO RNAs, microRNAs, and so on and so forth. Okay. And so what we perform actually before doing the fragmentation and reverse transcription, it's called enrichment. Okay. So we are basically enriching our total from our total RNA, the RNA of interest. Okay, and most of the times, I would say like 90% of the times, it's a messenger RNA. Okay, of course, there are like some applications that require also non-coding RNAs. It depends on your lab, or whether your lab is focused on that aspect as well. But most of the times, it's a messenger RNA. And so we're taking a look at these slide right here. These are the two main enrichment procedures that are performed into RNA-seq library preparation. And so let's start from this one on the right. It's called, wow, my mouse is just going crazy. All right, poly-A enrichment. So what is going on here is we have our RNA that has been extracted. Okay, just gonna stop using the mouse. Basically, we are mixing our total RNA with these magnetic beads. Okay, so you see this brownish circle, that's a magnetic bead. And these are modified with a stretch of oligo-DTs. Okay, and those oligo-DTs are able to recognize the poly-A sequence of our messenger RNA. And so we are able to capture, after some washes, only the fraction that it's a poly-A positive. Okay, so if you're interested only in messenger RNA, this is the way to go. Okay, so you perform a poly enrichment. Okay, In this case, you get rid of everything that you have, everything else that you have in the mixture. So you get rid of the ribosomal RNA. Okay, you get also rid of. Let me change color. You get also rid of transfer RNAs, long non-coding RNAs, SNO RNAs, pseudogenes, microRNAs. Okay. However, for some applications, you're interested in both the messenger RNA and the long non-coding RNA, for example. Okay. So if you want to have the information from these two species of RNA, what do? Well, you perform something that we call a ribosomal RNA removal. Okay. So if the poly-A enrichment is basically something that we are enriching our sample in, the RNA removal, it's more like a depletion type of approach. So I'm treating my total RNA with some reagents that depletes from the total RNA the ribosomal RNA. Okay. In this case, I'm not enriching for messenger RNA and long non-coding RNA. I'm just getting rid of the ribosomal RNA. So I will still have tRNAs, SNO RNAs, pseudogenes, and microRNA in my solution, although both microRNAs and transfer RNAs will not be incorporated into the library preparation um, um, because of the steps that we're going to do next. And so this type of depletion, there are like two main protocols for that. One is Ribo0, that's the very first one that has been used. And you basically have probes that are complementary to the ribosomal RNA sequence. And then those probes, you basically use them. They are biotinylated with a magnetic bead. And then you're able to basically remove the ribosomal RNA that it's bound to the beads, and everything that it's in the supernatant is basically not ribosomal RNA. Or the other approach, which is cheaper in terms of cost of reagents, is this RNase-H approach. For example, so Ribo0 is the approach that Illumina historically used, and RNase-H, the approach that both Kappa and NEB use right now. What they do is they basically mix your total RNA with these oligos. There are DNA oligos. And then if you treat this mixture of DNA and RNA oligos with RNase-H, which is a RNase enzyme that recognizes DNA and RNA hybrids, it will degrade only the RNA that it's bound to DNA. Okay, so if the DNA oligos that we have are recognizing only the ribosomal RNA, this enzyme will only degrade the ribosomal RNA and leaving the messenger RNA intact. Okay. Again, you not only have the messenger RNA, you also have all the other non-coding RNAs. Okay. So again, this is more like a type of depletion versus the enrichment of the poly-A approach. Okay. So okay, we've done enrichment. Okay, so what do? Okay. After enrichment, we usually fragment the RNA. If you remember from day one, from yesterday, actually, I briefly talked about a method to chemically fragment the RNA. And this is basically using a buffer that has magnesium or zinc. And you basically add this buffer and then you heat it up, usually like between 80 to 94 degrees, depending on the size of the RNA that you want. And you're gonna have fragmentation, okay? After you have fragmented your RNA, because it's already enriched, so everything that you have in solution is what you want, okay? You do fragmentation, you do random hexamer binding, okay? And this is the primer for the reverse transcriptase to produce the first strand of the cDNA, okay? So we're using this enzyme with the random hexamer as a primer to just produce cDNA, okay? The reason why we do fragmentation before, it's because most of the times, I mean, the reverse transcriptase enzyme is not a very efficient enzyme, okay? So it has troubles just, just reverse transcribing the full length of the transcript, which are usually like hundreds of bases to kilobases. And so this enzyme is much more efficient on shorter templates. Okay, so after performing the reverse transcriptase, we transform our single-stranded. Okay, so the output of this reaction is a single-stranded DNA. Okay, we are transforming our single-stranded DNA into a double-stranded DNA. And we do this by performing the so-called second-strand synthesis, okay? And this is actually a reaction that requires quite a lot of enzymes, okay? So it requires a DNA polymerase, it requires an RNase-H, so the same enzyme that we saw before. In this case, the RNase-H is degrading the RNA that was still complementary to the single-stranded DNA that the reverse transcriptase has done, and a DNA ligase, okay? So what the RNase-H is doing is basically degrading most of the RNA, but some of the RNA will still be there, okay? And the RNA that is still complementary to the single-stranded DNA can act as a primer for the DNA polymerase in order to create the second-strand synthesis. And so what you basically end up with, it's short fragments of second-stranded DNA on the first strand. So here's the first strand. Maybe I have a finer, yeah, first strand. And this is a second strand. You see you have these little pieces of DNA, and the DNA ligase is actually linking these all together, okay? So what is the output of these two reactions, okay? I basically have short, okay? I've done fragmentation already, okay? Plus double-stranded DNA, okay? And that is actually the same material as the regular standard DNA lab preparation after sonication, okay? So what I do is easy: N repair, phosphorylation, A-tailing, adapter ligation, and then PCR, okay? So at this point, once I have a short double-stranded DNA, then I can basically perform exactly the same steps as I performed before in the DNA lab preparation, okay? So far, I hope everything is clear. I'm adding a new information here, which is, I think it's really important. So the protocol that I described you is kind of like a old school. Nobody uses it anymore. Now everybody has switched to this stranded RNA protocol. The idea is exactly the same. There's just one little modification that you can see highlighted here in yellow on the right. During the second strand cDNA synthesis, we are adding UTP in our mix, okay? So I forgot to mention that, of course, whenever we are doing second strand synthesis, we also have nucleotides, okay? Otherwise, the polymerase will not be able to add anything, okay? So we have nucleotides here. And in this case, the mixture of nucleotides is actually different. It's just this little difference will make a huge improvement in the data that you have. Okay, so the DNTPs that we have here, they're not the canonical TCGA, but they're actually UCAG. Okay, this UTP that we're adding is still the oxynucleotide, so it's still like a DNA UTP kind of nucleotide, okay? So it can still be incorporated by DNA polymerases, okay? And so the second strand will basically be labeled with a lot of uracils in the sequence, okay? So we perform this special second strand synthesis, but then we still have our double-stranded DNA, okay? That is short, okay? So then we can perform N repair, phosphorylation, A-tailing, and adapter ligation, okay? So what happens next, okay? what happens next is that one of the two filament has still these uracils, okay? So this is the filament that was derived from the second strand. At this step, after the adaptor ligation, we have an incubation with an enzyme that is basically degrading the filament with the uracils, okay? So if this enzyme, which is called basically user, okay, so it's an enzyme that it's commercialized by NEB, it's a mixture of enzymes actually commercialized by NEB, is able to basically degrade the filament with the uracil that is containing the uracil, okay? So this is one approach, okay? And the second approach, an alternative, is using a DNA polymerase that is not able to read the filament with the uracil, okay? So if you remember from previous slide, when we were talking about DNA methylation, after you treat the DNA with bisulfate, you have your fragments that contain uracil, and then you need a polymerase that is able to read uracil in order to be able to amplify it. In this case, we're actually doing the opposite. We're using a polymerase that is not able to amplify, is not able to recognize uracil in the template, and so we're not able to amplify this filament. But we are able to amplify the other filament, okay? So this filament is okay, and it can be amplified. The other filament is not, okay? The other filament will not be amplified. So, okay, but so what is the point? So why am I doing this entire process if I'm just getting rid of one of the two filaments then, okay? And the reason is, I'm just trying to explain it here, okay? So we're trying to draw a cartoon of what's actually happening here, okay? So this is an RNA, it's 5' to 3', okay? And then we have our first strand synthesis, okay? So this is done with the reverse transcriptase, okay? 5', 3'. And then we do second strand synthesis, okay? So remember, here is 5', 3'. And our second strand synthesis, it's going 5' here to 3' here, and it contains uracil, okay? So after adaptor ligation, okay, I have my second filament up top with read 1 here and read 2 here, okay? However, this filament gets degraded, okay? So I don't have this anymore because either I'm not able to amplify it or I digest it using this user enzyme. So what I'm left with is this at the bottom, okay? So this at the bottom is actually the same as the first strand, okay? But if you compare what your output is, okay, which is this one at the bottom, you see is actually the reverse complement of the original RNA, okay? So if your original RNA started from 5' to 3', what you have here is from 5' to 3'. So it's exactly the reverse complement, okay? Just because your read 1 adapter ends up always on the opposite side of the 5' end of your original RNA, okay? You can try to draw it yourself, and you'll see that when you have a stranded type of library preparation, the read 1 is always giving you the reverse complement of the original strand of the RNA, okay? If you had unstranded library, then you will also retain these second strand, you will also retain this second strand filament. And in that case, your read 1 will give you both the same filament or the reverse complement filament, okay? So you're not able to distinguish where the original RNA was coming from, okay? And this is particularly useful for, especially if you're studying viruses, okay? Viruses have a way to basically just compact their genome and they're usually able to transcribe from both filaments, okay? And so the ability to check the strandedness or the origin of the strand of the RNA, it's really, really useful, okay? It happens in humans as well. So it's, I think now it's, you know, everybody's has switched to this stranded RNA. It's just the same protocol. It's just you have much more data, much more information coming out of the protocol. So I'll suggest you to use this one for your next preparation. Okay. All right. Let's check if there are some questions about this. I'll give you a couple minutes. Okay, there might be a question. Let's see. Once you're done, please move your selected cell so I can see your question. It's probably just hanging there because I usually should be able to see it dark gray. Let's see. Okay. All right. Hopefully, yeah. Okay. So probably no questions. Sounds good. All right. So we can just go ahead. Okay. All right. Quick question. Are you able to see if I highlight on my presentation like this, or? Oh, okay, perfect. Yeah, okay, sounds good. All right, so what I told you so far is actually partially right, okay? You remember the slide that we talked about the enrichment, okay? So if you want to study messenger RNA, you always do poly DT selection. If you want to study something else, you do ribosomal depletion. Is actually not always true, okay? So in this section of the workshop, I'm going to show you why the quality of your RNA actually determines the type of library preparation that you're going to perform, okay? So in this slide right here, you can see the difference between intact RNA and fragmented RNA. So we are pretending that we have fragmented RNA after extraction. Okay, so after extracting your RNA, you have fragmented RNA versus intact RNA. This happens usually. So if you have like a cell cultures, usually extract fresh RNA, and it's usually this type of RNA that you have. If you extract RNA from tissues that maybe were stored like a -80 for like a long time, you might have problems with the fragmentation. Also, FFPE samples, they're usually very fragmented, like highly fragmented. And so you can see, for example, microRNAs, they're like so short, it's basically impossible to notice a fragmentation pattern. Long non-coding RNAs can be fragmented, messenger RNA can be fragmented, and in this case, this is really important, okay? So if you have an intact messenger RNA, you see that the 5' end, where you have your cap, it's basically on the same molecule as the poly A tail, okay? But if it's fragmented, you see your 5' end cap here, that it's not on the same molecule of the poly A tail, okay? And this is making like a huge difference when we are trying to study messenger RNA in fragmented samples, okay? I'm gonna explain that in more detail later on, okay? But how do we assess the quality or the integrity of our RNA, okay? So here you see some images, okay? So this is a TapeStation image. You know, you can think of it as an agarose gel, fancy agarose gel. Do not take into consideration this green bar at the bottom. That's just a lower marker. That I'm going to explain that to you tomorrow. We have 2 RNA ladders here. They're indicated as RNA ladder. And then we have different samples, okay? So they're labeled A, B, C, G, R, S, okay? So I'm asking you, which one do you think has the best quality of all? A, E, or R? And you can type in your answer in the chat. It's just like a, try to guess. You don't know the answer. I didn't explain the answer. We're gonna um, um, all right, perfect. Yes. So A has the best quality. Okay. Um, why is that? You see these two peaks. Okay. These are actually question. Do you know what these two peaks might be? So this is a total RNA. Okay. So I'm loading total RNA. Great. Ribosomal RNA. Okay. Um, if you remember, the ribosomal RNA is the most abundant, okay, and luckily there are like a very few molecules of ribosomal RNA in terms of like species, and two of them, for mammalian cells, for example, are 18S and 20S, and these are exactly the ribosomal RNA peaks that you see here. Okay, so the entire study of the integrity of the RNA is actually based on ribosomal RNA, and we see this. For example, the TapeStation and the bioanalyzer, they have these so-called RIN score, which stands for RNA integrity number. And it's basically a number between 1 and 10, with 1 being very degraded and 10 being very intact, okay? So as you can see, which ones were the A, E, and R? So you can see A has 6.9, E has 4.4, and R has 2.5. So you can definitely see that A is better than E, and E is better than R. Okay, and how does the TapeStation or the bioanalyzer measures these number? Okay, so it's looking at the 18S and 20S peaks. Okay, so just look on the right. It's also looking at the area of 18S and 20S. Okay, so I think the best area has to be bigger than 2. So 2 times 20S compared to 18S of the area under the peak. And then it's also looking in this fast region. Okay, so the messenger RNA, actually they're like so minimal in terms of like percentage of total RNAs, like 5%, and they also have different sizes. Okay, so you will not be able to see a peak of messenger RNA. You will not be able to see something like this. Oh, this is messenger RNA. That's not possible. Okay, they all have different sizes. Okay, so that's why you don't see anything. You might see like a little smear like this, but it's very difficult to see and to distinguish it from the background. Also in the fast region, which is basically between the 18S and the 5S, which is another ribosomal RNA molecule that it's much smaller in size. If you see some RNA or some signal in this region, it's most likely coming from degradation of these two peaks. Okay, most of the times when you see some material in the fast region and you still see peaks, usually the ratio is not 2 to 1. It's much smaller. So most of the times the 20S might be degraded, and so the ratio is not 2 to 1, it might be lower. And then the degraded 20S, you see it as a smear. Let me change color. You see it as a smear in this region right here. Okay, in the so-called fast region. Okay, so let's take a look at our data here on the left. Okay, you see, so whenever you see two distinct peaks, 20S and 18S, that's a good sign. For example, for C, let's look at C. Okay, and compare it to, for example, D, that it's next to it. Okay, so in both samples, you see that you have the peaks. If you notice the intensity of the peaks, you see that in sample, where are you? In sample D, the 20S, which is this one up top, is much darker than 18S. That's what it's supposed to be. Okay, so the ratio has to be bigger than 2 to 1. In case of C, you see that actually the 20S is not as dark as the 18S. And what else do you notice? This area right here, it's much darker than the area right here. Guess what this area is? It's the fast region, okay? So basically the degraded 20S is running now here and here in the fast region, okay? And it's very different from the sample D, which has a very high RIN score, okay? And then we look at E, for example. It's barely impossible to see a peak of the 20S. It might be all degraded, and you see here you have a much darker fast region. F is good, G is better than E but worse than F, and everything is, you know, reflects the RIN score that you see at the bottom. And then you go to R. R is absolutely degraded, okay? There's no 820S peak. There might be a small 18S peak, but it's you know, it's difficult to say. And then you have a huge amount of RNA in the fast region, okay? Okay, so if we have these results, then everything actually changes, okay? So can you do poly-A if you're interested in messenger RNA in the case of R? The answer is no. If you remember what I told you before, that if you have degraded RNA, the poly-A is not covalently linked to the 5' end of your messenger RNA. Okay, so these are two distinct molecules. So if I'm pulling the messenger RNA using oligo-DT, okay, so which is complementary to this region, I can only capture this part of the transcript, but I'm not capturing this. I'm not capturing this. Okay, so what I'm left with, it's the very short fragment that it's close to the poly-A tail. Okay, so that's not a good choice if I have a very degraded RNA. Even if I'm interested in only messenger RNA, the approach that I would take is ribosomal RNA. Okay, if you remember, ribosomal RNA is not an enrichment; it's a depletion. Okay, so I'm depleting from the total RNA sample my ribosomal RNA, which is the most abundant molecule, and then I have in solution everything else. Okay, fragmented mRNA, good, I have it. Okay, so I can prepare a library from that, and I can sequence the entire length of my transcript. Okay, so it's pretty easy to say, okay, so if I have a RIN score below 5, then I'm going to do ribosomal RNA. But if my RIN score is you know, very high, I can do, you know, whatever I want. If I'm interested in messenger RNA, I can do a poly-A. If I'm interested in messenger RNA and long non-coding RNAs, for example, I can do ribosomal depletion. How about in the middle here? Okay, how about these numbers? 6.9, 6.4, 5.5. What would you do? Okay, so it's still possible to do a poly-A. The only thing is you expect to have the so-called 3' end bias. Okay, so your reads are coming mainly from the 3' end of the fragment. Okay, you know, doing poly-A is faster. It's much cheaper, both on the library preparation side and on the sequencing side. So, you know, if you don't have that much money and you still want to do poly-A and your RNA is, you know, not great, but, you know, it's, you know, in between good and bad, you can still do poly-A. It's just like, you know, the quality of your RNA will be reflected in your final libraries. Okay. There are many approaches that are doing 3' end sequencing on purpose, just to basically save money on the sequencing costs. So it's still possible to perform a poly-A capture on fragmented RNA, but when it's very fragmented, then, you know, you're just capturing the poly-A tail and nothing else. So you're not going to sequence anything else except for the poly-A tail when you have a RIN score of 2.5 or around 4. Okay. And this is another view of a RIN score of very good RIN score. This was extracted from fresh cultured cells, and you see it's quite good. And this was actually extracted from FFPE samples, um, and you see the RIN score is very, very low. When the RIN score is very, very low, we are actually using in the lab a different metrics to test whether the library, the RNA, sorry, is even usable for RNA library preparation. Okay, so this method is called DV200. Okay, so it's not the RIN score anymore, and it's mainly used for degraded samples. So what that means is basically I'm checking how much of my material it's above 200 base pairs and how much of my material it's below 200 base pairs. Okay, so if the material that it's above 200 base pairs, it's high, so it's like more than 70%, I can still go ahead and prepare it. When it's medium, I can still go ahead and prepare it. So if it's above 50%, I can go ahead and prepare it, even if the quality in this case is going to be quite bad. If it's low, in terms of like below 50% or too degraded, you can still perform the library preparation. It's just the results that you're going to get out of the sequencing is going to be pretty bad. Okay. So from a technical point of view, you can still prepare libraries, just the sequencing results are not worth probably the investment. Okay. Using the method that I've described so far. Okay. So for example, in this case, the RIN score is 2.3. The percentage of the sample that it's above 200, it's between 55 and 60%. So it's in the medium range, so you can still prepare the library. Okay, the results will not be great, but you can still prepare a library. Okay, and I'll give you an example. So I prepared a library using these exact RNA, and then we'll discuss later what is the best approach for it. Okay, so one of the two methods that we already discussed today, it's called ribosomal depletion. So I prepared my sample using ribosomal depletion type of approach. However, the results were not great. And so that's why I switched to a different type of library preparation that we're going to describe in the next slide. So this approach is a targeted approach. So you remember yesterday that we discussed the targeted DNA application, and if you remember from yesterday, we used the targeted DNA approach to introduce the UMI because we needed to perform a lot of additional PCR cycles, and the idea is exactly the same. Okay, so what we're doing is we're preparing a library from total RNA. And what we're doing is we are mixing our total RNA library preparation with the biotin-related targeted probes, and the probes are then hybridized to our target molecules. And the streptavidin, which binds biotin-related probes, is able to create this ternary complex of my target RNA, my biotin-related probe, and magnetic streptavidin beads. And now I'm able to enrich for my sample of interest. Okay. I performed the library preparation with ribosomal depletion, the RNA sample that we were discussing before of the RIN score 2.3, and also with the targeted approach. And the targeted approach gave me like much better results. Okay. Unfortunately, when you're doing ribosomal depletion on a very degraded RNA, you might have a very huge background of RNA that is coming from everywhere, basically. Okay, so the results will not be great. In the case of the targeted approach, I have a probes that are targeting all the exons in the genome. Okay, so it's very similar to exome-seq. It's just the exome-seq, it's usually performed on DNA, while the RNA exome-seq type of approach is performed on RNA libraries. Okay, and the targeted focuses your sequencing results on the exons only. Okay, the advantage is that you don't need that many reads you need fewer reads than the ribosomal depletion approach. However, it's more expensive in terms of library preparation, okay? Because you have to perform this hybridization, which is usually quite expensive to perform, okay? But I'm telling you, it's worth the money because the data that you get out of it, is actually much better, especially when your sample is degraded, okay? All right, let's do a Kahoot. So same as yesterday, we're going to kahoot.it. So just log in with your devices, and then we are classic. All right. The pin is sorry. Here. All right. The pin is this guy. Let's see. So if you remember from yesterday, you have to be correct first and then as fast as possible. Okay. Just gonna wait one more minute so people can log in. And then we can just get started. All right. Everybody's logged in, or most of you are logged in. All right, let's get started then. Okay. All right. First question: what is the most abundant RNA species usually present in cells? Well, you've answered that already. So if you were listening, you should be able to answer this question. Ooh. All right. So the most abundant RNA species is ribosomal RNA. Okay, so not a messenger RNA. Messenger RNA are usually between like 3 to 7%, while ribosomal RNAs are usually above 70% in your cell. Okay. All right. Very good. Second question: which step is usually performed first during a standard RNA library preparation? Reverse transcription, PCR, adapter ligation, second strand synthesis, okay? There's actually a step that we usually perform before, but among those choices, okay? And the first step that we perform in this case, it's reverse transcription, okay? PCR is at the very end. Second strand synthesis is after reverse transcription because reverse transcription is creating the first strand. And adapter ligation is actually after the second strand synthesis, right before the PCR, okay? Last question, okay? All right, winged iguana is still leading, okay? All right, last question. Stranded RNA-seq, what is the difference compared to an unstranded library preparation, okay? A stranded RNA-seq library can only be sequenced per end. Introduction of DUTPs in the second strand synthesis. Fragmentation step is longer for the second strand stranded RNA library preparation. The adapters used in the adapter ligation are different between the two preparations, okay? Only one is correct, okay? A few more seconds so everybody can answer. And the correct answer is we are introducing DUTPs, okay? So the adapters are exactly the same between the two preparations. We are introducing DUTPs in the second strand synthesis so we can get rid of the second strand during either the either the PCR or before the PCR using this enzyme that degrades the DUTP containing strand. All right, so let's see. Mighty Ant ended up in third position. Soaring Owl second position, and first one. See, all right. So he won. He won at one. All right. Good job. Good job. All right. Let's go back to the serious stuff. Um, and let's see. Let's do like a quick 5-minute break. Okay. So we reconvene at 3:21. So 5-minute break. Okay. So you can just stretch your legs, and then we have two more topics, basically some specialty RNA-seq preparations and the best practices NGS. Okay. 3:21. Sounds good. All right. Let's start. So we're going to take a quick look at these different type of library preparation, especially when we have like a low input material. And low input material can be, single cells or a very limited sorted cell population of your interest. Okay. And so let's start with a low input type of library preparation. So how do we perform a low input library preparation? We usually do not. So let's say we are interested in messenger RNA. Okay, so most of the times you are interested only in messenger RNA. That's what we are focusing our presentation on. Instead of doing oligo DT magnetic beads capture, which it's quite efficient in terms of like removing everything else, we do like a lot of washes, and it's a very lossy procedure. Okay, so what we actually do is we perform we still use oligo DT, but in this case these are primers for the reverse transcription. Okay, so we can perform this reverse transcription reaction either on extracted RNA or on cell lysates. And then we have this oligo DT, you perform reverse transcription, so it's copying the first strand here. And then reverse transcriptases, they have a very peculiar feature. They will add 3 cytosines. So when they arrive at the very end of their transcript, they're just adding 3 cytosine, okay, in a row. In the presence of an oligonucleotide that we call TSO, which stands for template switch oligo, TSO, which has a terminal GGG sequence, then the reverse transcriptase basically sees this pairing between the TSO and the 3 cytosines that you just added, and it has this template switch mechanism. Okay, so if up to here, it was doing reverse transcription, so from a substrate of RNA, so this is an RNA substrate. Okay, it switches, so that's the origin of the word switch. It switches template type. Okay, the TSO oligo, it's a DNA oligo. Okay, so from RNA here, it switches to DNA oligo here, and now it basically copies the information. And the result of this is you have a primer handle in green here, and another primer handle that I just highlighted here at both ends of the transcript. Okay, I just want to mention this is a full length transcript. Okay, so you're doing reverse transcription on the full length RNA. What you do next, you do PCR amplification. Remember, we are starting with a low amount of RNA, so we do like a PCR amplification, and then we have our cDNA library. Okay. However, as I told you, this is a full length reverse transcription, so these fragments can be, even kilobases long. Okay, so up to 2-5 kilobases usually. And so it's too long to be sequenced on an Illumina sequencing machine. So what do? Is we use this fragmentation approach that I described yesterday to fragment and attach adapters at the same time, okay? So once I do this, I basically have my reverse transcribed double-stranded DNA with the adapters at the end, okay, these blue and the red. And then I do PCR amplification and I introduce the barcodes or the indices and the P5 and P7 sequences that are necessary for sequencing on the Illumina machine, okay? So this is the approach, this is the most used, or the original approach that a lot of people use for low input RNA or even single cell, okay? Yeah, so it's based on the template switch property of RT enzymes, okay? What else? Okay, so what if the RNA that I have is low and degraded, okay? What you can think of is, okay, so if it's degraded, I'm gonna do ribosomal RNA removal, okay? Also, the RNA ribosomal removal, it's lossy in terms of procedure because you have to take the supernatant, you have to precipitate it, or you have to clean it up. So it's very lossy, okay? And remember, here we have a very limited amount of RNA, so you don't want to lose your RNA during this step. So what this library preparation, this is from, one type is from commercialized by Nugen, the other one is commercialized by Takara, okay? But the idea, I mean, the steps are different. I'm not gonna go too much into details, but the idea is the same. We are removing the ribosomal RNA derived reads at the end of the library preparation where we have a huge amount of material, okay? So we've done PCR amplification, so we've increased our amount of library. And so now, even if it's a lossy procedure, we can get rid of the ribosomal RNA, we have a lot of material, okay? Nugen is doing this by using a restriction enzymes that basically recognizes the adapter to ribosomal RNA junction, while Takara is using CRISPR-Cas9 in vitro, so like in a tube, to basically just degrade the DNA that is, annealing to the guide RNA against the ribosomal RNA reads, okay? So two different approaches. The strategy is exactly the same. Ribosomal RNA removal at the end of the library after we've done PCR amplification. So it's still possible to perform a library from degraded low input RNA if your material is precious, okay? Other methods of amplification. So far you've seen, we've done, whenever you see library amplification here, and also here we do like a PCR. So these are all PCR steps, okay? So it's an exponential amplification of your material, which can introduce a lot of biases. There's another approach, which was first introduced by this Methyl-seq method, that it's usually, it's using in vitro transcription instead of PCR, okay? So in vitro transcription has, it's producing a lot of output, but it's a linear amplification instead of being a exponential amplification as the PCR. So the advantage of doing in vitro transcription instead of PCR is that you maintain the ratios between the RNA molecules that you had at the beginning, okay? So it's more like, it's introducing fewer biases, okay? The drawback of in vitro transcription is that this in vitro transcription is producing RNA, okay? So you have to transform the RNA back to DNA. So what you're doing is actually RNA, DNA, okay? And then you are doing in vitro transcription to just increase the amount. So you have RNA at this point, and then you have to go back and transform it to DNA before sequencing, okay? So it's more like a ping pong or back and forth type of strategy. But again, the advantage is maintaining the linearity of the transcription, okay? All right. Before switching to single cell. Let me just quickly check if I have questions in the spreadsheet. Okay, no questions for now. It's good. Let's go ahead. Um, um, so here, so the previous approaches, some of them were also available for single cell, but some others were only for low input. For single cell sequencing, there are like different approaches. The first one that was developed as a high throughput approach was the so-called DropSeq. Okay, so was what the DropSeq does, and you know it's somehow similar to what all the other commercial technologies do, especially for 10x and Illumina Biorad. They're basically able to encapsulate in a droplet of aqueous solution in in an oil emulsion both a single cell and the reagents that are needed for reverse transcription. Okay, on top of the reagents that are needed for reverse transcription, you also have these beads, and these beads, they have barcodes. Okay, and the barcodes that are present on the beads are specific for each cell. Okay, so each bead will have a barcode that is different barcodes compared to different beads. Okay, you see here you have orange, you have light blue here, you have what do you have there, yellowish maybe. So they're basically just different barcodes. After you perform the reverse transcription, and at this point you've basically introduced the barcode in your first strand, then you basically mix everything up, and then you perform PCR amplification. And so after you've done sequencing, you can say, oh, this read is coming from this cell because it has this barcode. This read is coming from this cell because it has this barcode, and so on and so forth. Okay, so these are different from the barcodes that we've seen before on the adapters. These are just different barcodes. These are cell barcodes, not sample barcodes. But these are giving you the information of what is the origin of the fragment. What is the origin of this transcript? What cell was it coming from? Okay, so you have different origins. Okay. Another approach by BD, it's basically, instead of using this oil emulsion type of approach, it's using microwells. Okay, so the idea of BD is just like flowing a suspension of single cells onto a microwells, and then only one cell per microwell will be captured. And so you have thousands and thousands and thousands of these microwells into one cartridge, which is it's not depicted here. So you can capture, tens of thousands of cells but the idea is not oil emulsion like in the case of DropSeq and 10x. It's more, gravity type of approach. Okay, so only one cell will go into each well. Okay. I have to say the leader in the market as of now is 10x Genomics. It does not only have a single cell RNA platform. You can also do single cell DNA if you want to check for mutations, especially in cancer. If you want to sequence the DNA of a biopsy, you can use this one to just basically understand what's the mutational load for each single cell. You can do RNA-seq. This is the approach that we described. And I just want to mention that this approach is only sequencing the 3' end of the transcript. You just want to see if a transcript is present or not. You don't care about the rest of the transcript. Okay. You can only do. You can also do VDJ, and this is called the 5' end sequencing. So it's RNA sequencing, but it's sequencing the 5' end. It's not sequencing the 3' end. So it's like the opposite approach. You can do Ataxic that we've seen before on single cells. Again, genome, exome, and also de novo assembly of new genomes. Okay, so it's a very good application. Also, they recently acquired these companies called Spatial Transcriptomics. So 10x is not only specializing in single cells, but it's also specializing in um, where the transcript was in a um, tissue biopsy. Okay, so this is the tissue biopsy. So you have a thin layer of tissue, and then you put it basically on these slides, and at each position in the slide, you have a barcode, and the barcode is telling you, okay, so this position was exactly in this part of the tissue, and so you're basically doing RNA-seq not only to measure the abundance in single cells, but also to understand where these cells were originally in the tissue that you in the tissue layer that you extracted. Okay. This is just a slide for BD. I told you before. You have microwells. Okay, so just one cell in each microwell on average, and but everything else is similar to 10x. Also, BD allows you to perform a targeted approach. So it's a targeted RNA-seq, okay? While 10x is a full transcriptome. And they claim that the targeted RNA-seq is much better in resolution, especially for low abundance transcript. But, you know, that's their claim, okay? All these approaches that I told you about, they require an instrument, okay? So a specific instrument that does that. 10x as a, you know, an instrument for emulsion of cells and for the creation of those. And also BD has this instrument right here that basically allows you to deposit one cell per microcell. However, there are also other approaches that are not using any separation methods. So the 10x is using basically aqueous droplets in an oil for separation. BD is using microwells. In the case of this type of approach, which is called, you know, split sequencing, they still use the cells as, you know, physical method to distance the transcriptome from one cell to the other. Okay, so they have a specific protocol to just block the cells as they are, so they're not basically releasing their transcriptome, and then they're serially tagging the RNA molecules with barcodes, and just by pulling and splitting these cells will give you the combination of barcodes that identifies each single cell. Okay, and this method theoretically is able to analyze a lot of cells, and it's instrument free. Okay, it's just like a longer in terms of library preparation. Okay, but I just want to let you know that it's out there. All right, so a couple of slides on small RNA, and then we're almost done with the RNA part. So the small RNA, so the main difference is that we are starting with a very small fragment. Okay, so can you type in the chat if you know how long the you know, usually small RNAs are? Examples are microRNAs, small interfering RNAs, and stuff like that. So if you want to guess how many nucleotides a small RNA is. All right, sounds good. Yeah, they're usually between 18 to 30 nucleotides long, okay? And so this is a very, you know, it's a challenge, you know? It's very short, so we have to be able to capture a very short fragment, which is very different from messenger RNAs, which are usually hundreds or kilobases long, okay? So the library preparation that we're using in this way, it's totally different, okay? So the first thing that we do is actually we are ligating adapters on the RNA, okay? So we're using a specific two specific enzymes actually, RNA ligases 1 and 2, to ligate adapters at the 3' end and at the 5' end, okay? So once we do adapter ligation on RNA, we add a primer that it's complementary to the adapter at the 3' end, and then we perform reverse transcription, okay? And the reverse transcription is creating the first strand synthesis. And at this point, you have the adapters at both ends, okay? So you can go ahead and do a PCR right away. And the PCR usually introduces both indices, or just one index if it's a single index, and then the P5 and P7 sequences that you have, that you need to have for sequencing on the Illumina machine, okay? Question: if you have adapter dimers, do you think it's going to be easy or difficult to remove it in a small RNA type of library preparation? Okay, remember, in this case, the insert is only 20 nucleotides on average, so your adapter dimers will be 130, let's say 130, and then your full length product will be 150, 160 tops. Okay, you think it's going to be easy or difficult to separate the two? You can just type that in the chat. It's very difficult, okay? Very difficult, okay? So that's why with RNA-seq, a small RNA library preparation, most of the times you have to perform site selections on even on page gels, okay? So on polyacrylamide gel. You can do it before starting the preparation, so you just basically just get rid of all the other RNAs. You do it after the PCR, so at the very end, to just get rid of the adapter dimers, or you can actually do both at the beginning or at the end, okay? Whenever you're doing a gel extraction, you're losing a lot of material, okay? So unfortunately, this is, you know, a very difficult problem to solve. There are protocols that are, you know, they solve the issue just by using specific reagents, but still, you might have a huge contamination of adapter dimers in small RNA-seq libraries. Luckily for you, you don't need too many reads. We're talking about like, let's say 5 million reads for mammalian small RNA transcriptomes. And so even if you have a huge contamination of adapter dimers, you will not, you know, impact the sequencing costs a lot, okay? All right, so last slide of the RNA-seq, and I think it's one of the most important ones on top of the how to determine the quality. And then I'm gonna check for questions, and then we're gonna go to the final section of the workshop, day 2. Okay, so let's start from, why don't we just start from the right? Okay, so I'm interested in ribosomal RNA. First of all, I don't believe you, but if you're really interested in ribosomal RNA, you need to perform any sort of enrichment. 90% of your sample is coming from ribosomal RNA. So please go ahead, prepare your sample, either stranded or unstranded type of library preparation, okay? If you're interested in small RNA, then you only have the small RNA library preparation option for preparing the libraries, okay? So there's just one type of library preparation, okay? If you're interested in messenger RNA, now things become interesting, okay? Is your sample degraded, okay? No, my sample is not degraded. I can do poly-A enrichment. You know, it's cheap, it's easy, it's fast. Again, I suggest you to go ahead and perform a stranded type of library preparation, okay? Is your messenger RNA degraded? Yes or no? You can perform a targeted type of approach. If you remember, it's more or less the same as the exome sequencing. The exome sequencing that you know, it's usually starts from genomic DNA, but you can perform a similar type of approach on RNA. Or if you're not interested in, you know, the entire transcriptome, maybe you're interested only in genes that you know are overexpressed in a particular type of cancer. You may have, you know, a breast cancer panel. You might have a colorectal cancer panel, and please do the stranded preparation. Okay. This is saving you money for the sequencing, is more expensive for the library preparation when I'm talking about the targeted approach. Is your sample degraded? Yes. Again, you can go either with the targeted or with the ribosomal RNA depletion. And this is only the strategy that you may want to utilize when you're interested both in poly-A positive and poly-A negative transcripts, okay? As long as they are not a ribosomal RNA, RNAs, okay? And then you perform ribosomal depletion. Please do this stranded type of library preparation, okay? Also, if you have low input, you can perform a poly-A enrichment, and then you can go down to single cells. You can do the targeted. Unfortunately, the single cell targeted, it's possible to do it with the BD type of approach, but with the targeted approach that we described for regular RNAs, it still requires a few nanograms of material. For single cells, can you perform RNA depletion? Actually, yes, I have to update this one. You can perform, I've done it. You can perform single cells RNA-seq using a ribosomal RNA depletion. So if you have single cells that you're interested in not only in poly-A, but also in other transcripts, then you can do that, okay? We are stopping until 3:55. I'm gonna check questions. And if there are any, and then you can stretch and stay hydrated, so you have like a few minutes. Let me go and check my screen. All right. If you don't have any questions, you can just step away, and we'll reconvene in 7 minutes, 7-8 minutes. Okay, welcome back. As you can see on this slide, you also have other workshops that are related to mine. Again, workshop 16, my workshop is more dealing with the experimental type of NGS, while these other workshops, they are more focused on the analysis, okay? So if you go to the collaboratory website, you can you're able to see all these workshops that are still, yeah, they're all of them are still happening, okay? So you have time to register, okay? So let's go to the last section, and then we're gonna have like a very small quiz at the end, and then you're free to go, and we'll see you tomorrow. So the best practices in the NGS. So I think it's really important, and it's a duty for me that to just teach students the best way to prepare the libraries, so they can, you know, if they want to prepare libraries in their lab in the future, they know, you know, they're doing the right job, a good job. Okay. There are like 3 topics that I want to talk about. We're going to briefly mention the spike-ins. UMIs, we've seen them already. I just want to stress that out. And then the very important UDI adapters. Okay. So spike-ins. Usually the main assumption whenever you're analyzing data, the main assumption that you do with the quantitative data, like for example, ChIP-seq or RNA sequencing, is that the total amount, let's take RNA-seq as an example, the total amount of transcripts that you have in each cell is exactly the same, okay? So let's say you have 20,000 transcripts in this cell, even if this cell is treated with, you know, like a kinase inhibitor or something like that, you still have, the assumption is you still have 20,000 transcripts in that cell, okay? In terms of molecules that are expressed, okay? However, it's not always true. And so you should add spike-ins during the extraction, okay? During the extraction of the RNA to basically check whether this assumption that you're making is correct or not. Okay, there are like several spikings that are available. I mentioned also for ChIP-seq, for DNA sequencing, you know, virtually for everything, you know. And so these I think the paper that I put here, it's a very good one. So you can just if you're interested, just read it. And for example, if you're interested in RNA sequencing, they will have the list for RNA spikings and which one is better and so on and so forth. So they're basically discussing not only why you need a spike-in, but also they're telling you which spike-ins to use. Okay. All right, UMI. Okay, so again, it's different from sample barcodes. Okay, so the difference between sample barcodes and UMI will be in the final test tomorrow, for sure. So please, if you have questions, just ask me questions about UMI and sample barcodes. I've seen from yesterday that in the mini quiz, some of you guys made mistakes between UMI and sample barcodes. So please, I just want you to I don't care about the results of the quiz. I just want you to understand the difference. Okay. So UMIs are tagging each individual molecule. Okay, independently of the origin. Okay, so if you look at the top right, sample one. Okay, all the index and barcode sequences have the same color. So the index and barcode sequencing is the same for all molecules coming from sample one. If you go to sample two, then all the barcode sequences will be different, but same within sample two. Sample three, exactly the same. So it's yellow. So it's orange, gray, yellow, green. Okay, so index and sample barcode are identifying the fragments coming from one or another sample. Okay, the barcodes, sorry, the UMI, my bad. As you can see, they're all different. You have green, red, yellow, green, red, red, green, red, green, yellow, yellow, yellow. Okay, so they are just tagging individual molecule. Okay. Remember when we discussed the UMIs yesterday, the UMI sequence is not sufficient to say, oh, this is a PCR duplicate. We usually use two information, the mapping location and the UMI sequence, okay? So I cannot say that these three reads, since they have the same UMI, are coming exactly from the same original molecule, and so they're only PCR duplicates. I cannot say that, okay? It's possible that these three UMI and reads are mapping somewhere in the genome. So for example, read one here, read two here, read three here. And so if they are located in different parts of the genome, then they're not coming from the same molecule, obviously, okay? I just want to stress the difference between the index barcodes or UMI, okay? So please, if you are starting a new protocol, and I'm telling you, it's a very overlooked thing, but especially in RNA sequencing, where for some genes you have crazy coverage, and so it's possible that a lot of fragments are starting and ending at the same position. You know, we usually do not remove PCR duplicates in RNA-seq if we don't have the UMI, because it's possible that for some genes you have true biological, duplication. Since the coverage is so high, you can have reads that are starting and ending at the same position. Okay, so if you remove potentially PCR duplicates, you can remove true biological signal. Okay, so if you're starting a new RNA-seq project, and if you have the money to just buy adapters with UMI, please do so. Okay, it's very useful. It will give you a very good quantitative result of your RNA-seq. Okay. The other thing I want to mention, and this is more like everybody should start using this, it's the use of UDIs. Okay, so the indices that you can have in your adapters, they can be different. Okay, so if you look on the left, this is the configuration, the possible configuration of the barcodes and the adapters. And so when you see SI, so these are the so-called single indexed. Okay, so you only have one index. Okay, so just forget about index 2, you only have one index, and that's identified with SI. You can have the so-called CDI. Okay, so CDI stands for combinatorial dual index. Okay, so in this case you have two indices, but it's the combination that it's unique. Okay, so for example, sample 1 has blue black, that's one combination. Sample 2 has blue green, that's another combination which is unique, and sample 3 has yellow green, which is unique. Okay, but as you can see, for example, in index 2, the green is repeated between samples, and index 1, both sample 1 and sample 2 share the same index. However, if you're using the combination of the two, you can distinguish the molecules, whether they're coming from sample 1, 2, and 3. Okay. The other configuration is UDI. Okay, and UDI, it's, if you look at the schematic that I draw, it's quite simple. Okay, both index 1 and index 2 are unique to the sample. Okay, so sample 1 will have blue black, but nor blue nor black are repeated anywhere in the possible combination of the adapters. If you see, sample 2 is orange gray, and sample 3 is yellow and green. You don't see green anywhere else, you don't see yellow anywhere else. Orange and gray, you don't see them anywhere else except for their own sample. Okay, why is this important? We mentioned briefly yesterday this problem of the index hopping. Okay, and this is actually very, very important, especially if you're sequencing with the new sequencing machines, the HiSeq 4000 or the NovaSeq, because they have this so-called patterned flow cell. And the patterned flow cell basically is amenable to this type of problematics. So if you have small contamination of adapters or primers in your final library, they will be misincorporated into another library once you're doing the pooling for sequencing. Okay, and so if you have index hopping, one fragment, even if it was originally assigned, I mean, even if it was biologically originating from sample 1, it can be assigned by to sample 2. Sorry, one fragment can be misassigned from sample 1 to sample 2. Okay, you see they are sharing just one index. Okay, so if one fragment was coming from sample 1, okay, with index blue and black, and then the primer from sample 3, or the adapters from sample 3, the green one, is incorporated into sample 1, it gets swapped into sample 2, okay, because the green is substituting the black, and so now we have blue and green, which is a combination of sample 2 and not sample 1 anymore. Okay, so the fragment originally had blue black. However, with this index hopping problem, the black got swapped with the green, and now the combination that you have is green and blue, which is sample 2. Let's see what happens if this index hopping problem with UDIs. Okay, so the index hopping problem is happening independently of the type of the adapters that you have, of the configuration that you have. However, with the UDI, and we'll see an example in a second, you'll see that it will help you eliminate these identify and eliminate these index hop and misassigned reads. Okay, so let's go back to sample 1. So there's one fragment of DNA that is coming from sample 1, so it has a blue and black tag. Okay, now for example, a sample 3, the same adapter that was creating a contamination in the first example is going into sample 1, and it's swapping the black with the green, okay? So now you have basically a combination of blue, okay? And you don't have black anymore. The combination that you have is black, and let me find the color, black and green, okay? Okay, so I have my sample with black and green, sorry, with blue and green. Is this a possible combination? No, none of the samples have blue and green as a combination, okay? They have blue and black, they have orange and gray, they have yellow and green, okay? So when you see a blue and green combination, that is definitely an output of index hopping. But at this point, you can remove it, okay? So you're not misassigning the DNA fragment to a different sample. You're just removing the read because it's basically considered to be a contamination, okay? So I hope I didn't make too much confusion in here. I was playing with the colors. Sorry about that. And so UDIs is just giving you the advantage that you can recognize these, chimeric reads, and so you're able to discard them. The final configuration that you can have for NGS adapters, it's UDI, UMI. So UDI, same here. So you have unique, for each index, you have unique barcodes to distinguish the sample. You also have the UMI information that we've seen in the previous slide, okay? And for example, IDT, I usually order primers and adapters from IDT, but you can order from anywhere you want. They already have a preset custom service for adapters. Since the Illumina adapters are, universal. And so you can just go there and say, okay, I want a unique dual, or I want a unique dual with UMI. And so they will synthesize these adapters for you with all the right sequences and stuff like that. So you don't have to basically just create them yourself. Okay. All right. Let's check if there are questions in the Google spreadsheet. And then if there are none, we can go ahead with the final mini quiz. And then we are done for the day, and we'll start tomorrow with the library preparation online experience. Okay, so here's the link for the third mini quiz, and I will keep it open for 10 more minutes. So I'm just gonna stop it at 4:20, 4:25. And then I'll be here if you guys have questions. I'm here to answer the questions, and until 4:30, okay? Otherwise, please do the mini quiz. Again, it's required for attendance, and I'll see you tomorrow, all right?